Translation Research Centre from Patel Biotechnology and Bioinformatics Institute, Pune. The topic of his talk is Emerging Role of Long Non-Coding RNA in Oral Squamous Cell Carcinoma Progression and Therapy.
Good evening, everyone. First of all, I would like to thank for the organizing committee for providing me the opportunity. Myself, Dr. Subhayan, I am from Dr. D.Y. Patil Biotechnology Bioinformatics Institute, Pune. And the title of my talk is Emerging Role of Long Non-Coding RNA in Oral Squamous Cell Carcinoma Progression and Therapy. So this is the content of my talk. Now coming to the brief introduction. Many of us already know that the oral cancer is one of the most prevalent type of head and neck cancer, and that arises in the tongue, lips, oral cavity. Globally, oral squamous cell carcinoma or oral cancer is the 6th most common cancer, where India contributing 1/3 of the global burden. So this is the report. You can see that in India, around 77,000 new cases annually, with 52,000 deaths reported annually. So despite advancement of the treatment, the overall survival rate of oral cancer is around 50%, and even worse in case of the metastasis. On the other hand, long non-coding RNA or link RNA, LNC RNA, it is a type of non-coding RNA which are more than 200 nucleotide in length, and like mRNA, they are transcribed by the RNA polymerase II. So the link RNA interacts with the microRNA, thereby regulating the microRNA's interaction with the mRNA. Link RNA interaction either DNA or RNA or protein, thereby regulating cellular homeostasis. Currently, there are many evidences that report that association of the long non-coding RNA or link RNA in different types of cancers, including the oral cancer. And currently, the link RNA melatonin, hot air, CC81 are in clinical trials for the head and neck cancer, thyroid cancer, colorectal cancer diagnostic biomarker study. So as we are working on the oral cancer and on long non-coding RNA, so we wanted to know what are the candidate long non-coding RNA in the oral squamous cell carcinoma. So for this, you can see first we performed RNA sequence analysis using the oral cancer patient sample. And this is the differentially expressed gene data. You can see oral cancer versus adjacent non-tumor sample. And those are the differentially expressed gene, and the red color dot indicates that significantly differentially expressed gene. Using the significantly differentially expressed gene, we perform the gene ontology analysis and the cake pathway analysis. The gene ontology analysis and pathway analysis, you can see the most of the modulated pathways, that's metabolic process, cell cycle, PI3K AKT signaling pathway, RAS signaling pathway, those are modulated due to the long non-coding RNA in oral squamous cell carcinoma sample. From the differentially expressed gene, we identify one novel transcript, that is the EGFR long non-coding downstream RNA, ELDR. So it is one of the novel transcript. We were interested in two reasons, because the ELDR is upregulated, significantly upregulated in oral squamous cell carcinoma sample. This is the one reason. And second reason, you can see that ELDR gene is closely localized to the EGFR gene in chromosome 7 in the downstream and opposite strand. EGFR is one of the gene that is frequently amplified in oral cancer. So that's why we were interested the ELDR as it is closely localized. And there was no report before us, but we found significant upregulation of that gene in our RNA sequence data. We validated the data in oral cancer patient sample. So this is the data, and you can see most of the oral cancer patient sample, the ELDR is significantly upregulated. And the high expression of the ELDR is positively correlated with the cancer stages. This is the RNA fish analysis. You can see the ELDR localized both nucleus and cytoplasm in the oral cancer patient sample. To understand the molecular mechanism, we used the in vitro model. We selected two oral cancer cell line, Cal27 and JHU29, and we knocked down the ELDR gene using the siRNA. And first we check the cell proliferation, and you can see here the cell proliferation significantly downregulated when ELDR gene is knocked down. Next, we examine the EGFR expression. So I told you before that EGFR is closely localized to the ELDR gene. So that's why we thought, let's check the EGFR expression in the protein level first. So when we knock down the ELDR, we found that EGFR is significantly low. Next one is the rescue experiment. You can see when we overexpress the ELDR in the knockdown sample, the restoration of the EGFR expression. We next examine the EGFR downstream signaling pathway, STAT3 phosphorylation, and we found the STAT3 phosphorylation also inhibited. So what we observe, the ELDR regulate EGFR expression and it signaling in regulation of cell proliferation. Now coming to the mechanism, what would be the mechanism how ELDR regulate the EGFR? So we were three hypotheses. First one, the ELDR may directly regulate the EGFR expression by physical interaction. Second, ELDR may indirectly regulate the EGFR expression by some other interacting partner. And third, ELDR may indirectly regulate EGFR expression by microRNA. So for this, we perform the RNA pull down assay. So here we in vitro transcribe the ELDR RNA, which are the biotinylated ELDR RNA, and then we tag the ELDR RNA with the streptavidin magnetic bead. We incubate the RNA, the ELDR RNA with the oral cancer cell lysate and perform the pull down against the magnetic column. And we did the two experiments. One is the mass spectrometry, another is the Western blot. This is the mass spectrometry data where you can see the number of protein can bind physically with the ELDR. In that data, we did not see any physical interaction between EGFR and ELDR. But from the list, we found one interesting molecule, that is the ILF3, interleukin binding factor 3. So that molecule we found, it is the DNA and RNA binding protein that function as transcription activator and regulates mRNA stability. ILF3 is reported as to stabilize the EGFR and cyclin E1 to binding at the 3' UTR. So that's why we interested in ILF3. Let's check the ILF3. We verified the data by Western blot. This is the Western blot data, sense versus anti-strand strand, and you can see enrichment of the ILF3 in the with the ELDR sense strand. Next, we examine the consequence. What happened when ELDR and ILF3 interact? So Western blot data, using the Western blot analysis, we found ELDR doesn't regulate or does not change total protein expression of the ILF3. Rather, ELDR regulate ILF3 localization change. So if you can see the picture, you can see when ELDR is overexpressed, ILF3 express both nucleus and cytoplasm. When ELDR is knocked down, the ILF3 exclusively localized in the nucleus. So that indicates that ELDR sequestered ILF3 in cytoplasm, where ILF3 function different types of role. So first we examine whether ILF3 ELDR axis regulate EGFR expression or not. We knocked down the ILF3 here in oral cancer cell line, but surprisingly we did not see that EGFR protein expression change. So this indicates that ELDR ILF3 axis doesn't regulate the EGFR action, the EGFR regulation. But next we examine the, let's see the cyclin E1, another target of ILF3. Yes, we found, following knockdown of the ILF3, following knockdown of the ELDR, we found significant reduction of the cyclin E1 and its downstream RB phosphorylation, cell cycle analysis, G1S cell cycle arrest. we found. So from this data, we conclude that ELDR ILF3 axis doesn't have any role on EGFR protein expression change. Rather, it regulates the cyclin E1 expression change and the cell cycle regulation, cell cycle pathway regulation in oral squamous cell carcinoma sample. Now the question was how ELDR regulate the EGFR expression. So for this we examine few microRNA which were known to inhibit the EGFR expression. Among those, we found the microRNA 7 is a known microRNA that target the EGFR. So when we overexpress the ELDR, when we knock down the ELDR, we found microRNA 7 expression significantly up. When we overexpress the ELDR, the microRNA 7 expression significantly low. So that indicates that, but when we overexpress the microRNA 7, there is no change in the ELDR. So this indicates that ELDR inhibits microRNA 7, but microRNA 7 doesn't have any role in changing the ELDR expression. Again, when we overexpress the microRNA 7 in protein level, Western blot data, you can see the EGFR protein expression significantly low. This is the rescue experiment. We co-express the microRNA 7 and ELDR, EGFR expression restored. So finally we conclude that ELDR induce the EGFR expression through inhibition of the microRNA 7. So in conclusion, what we found, ELDR, the novel transcript, which is significantly upregulated in oral cancer sample, which regulate the cell proliferation. The mechanism is two. One, it induces the EGFR protein expression through inhibition of microRNA 7, and another is the ELDR IL-3 interaction induce the cyclin E1 signaling, resulting cell proliferation. So you already published that information. So next we wanted to know the ELDR is a novel transcript, whether it has any role to transform the normal oral cell or not. So we selected normal oral cell here, normal oral keratinocyte. It is immortalized cell. We made the ELDR overexpressed stable cell, and we examined the proliferation here. As compared to the wild type, you can see the ELDR overexpressed cell has significantly higher proliferation, colony formation, and in cell cycle we found significant change in G2M phase. We examined the histone phosphorylation, that is the G2M marker, and we found that ELDR overexpression significantly elevate the histone phosphorylation. To understand the underlying mechanism, we performed the RNA sequence analysis, and here we found the most of the genes upregulated which are G2M phase. This is the RNA data, and we found aurora kinase, PLK1, CDK1, cyclin B1, and their common transcription activator, FOXM1, all are upregulated. In the protein level, we already know that aurora kinase activate the PLK1 by phosphorylation. PLK1 activates CDC25C by phosphorylation. CDC25C is the phosphatase that dephosphorylates CDK1, thereby G2M progression. So in our Western blot data, we already show that the aurora kinase upregulated and CDK phosphorylation downregulated. To understand the mechanism, we looked back our RNA pull down data in the mass spectrometry data, and there we found another protein molecule, that is the CTCA, that interacts with the ELDR. So CTCA we selected because it is the known molecule that transcriptionally activate FOXM1. So that's why we selected the CTCF, and using the deletion construct, we identified that C-terminal domain of CTCF interacts with the ELDR. And ELDR CTCF interaction stabilizes the CTCA protein. We did the cyclohexamine mediated protein stabilization assay, and this is the immunofluorescence data. You can see CTCA expression is upregulated following ELDR expression, and when we knock down the CTCF, we found the FOXM1 downregulation, aurora kinase downregulation. This is the FOXM1 promoter activity assay. By this analysis, we found that the normal oral keratinocyte acquires selective growth advantage when we overexpress ELDR solely. And the mechanism is like that: ELDR interacts with the CTCF, thereby transcriptionally activate the FOXM1. aurora kinase signaling pathway and G2M signaling pathway. And this is my last slide. So third, we wanted to know the therapeutic efficacy of ELDR, and for this we used two oral cancer model system. One is the cell line derived xenograft model in nude mice. Another is the patient derived xenograft model, that is the PDX model in NSG mice. In both the cases, we injected the siRNA of ELDR intratumor injection, and we found that intratumor injection of the ELDR significantly reduced the tumor growth in both the model. So in conclusion, the link RNA ELDR plays important role in oral cancer progression, and targeting the gene has a potential therapeutic importance. So finally, I would like to acknowledge my current lab member, Ms. Ashpriya, Dr. Basu, Professor J.K. Pal from our institute. From our Bioinformatics Center, Dr. Dimple, Dr. Shuchi. From our dental college, Professor Supriya Kheur, Professor B.M. Ruragi. From our medical college, Professor Shomir Gupta. Professor Ratna Ray, she was my postdoc mentor, and most of the work I did under her guidance. She is from the St. Louis University, USA. And I would like to acknowledge DBT for providing me the Ramalingo Swami Re-Entry Fellowship. And thank you all for listening me. Thank you very much.